Good morning. It's March 26th, and this is your daily brief in science. Here's everything you need to know. A second study of 216 canid skeleton genomes redefines when dogs became our companions in Europe, suggesting that European Neolithic dogs, about 6,000 years old, descended from hunter-gatherer dogs dating back more than 14,000 years. The research implies domestication predates the agricultural shift in Europe and may have begun earlier in Asia, with puppies buried above human graves in Pinarbasi, illustrating a close, evolving relationship between early dogs and human communities. These findings point to dog evolution involving long-range migrations, interbreeding, and significant human influence as humans moved across broad regions. The News and Views piece, authored by Lauren M. Hennelly and Mikkel Holger S. Sinding, synthesizes and analyzes the new data, referencing related studies and citing the primary Nature articles and supplementary materials. The work sits within a broader genomic and paleoanthropological context, tying into prior research on ancient genomes and dog domestication, and emphasizes dogs' mobility and ongoing exchange with humans. Isotopic analysis shows Turkish dogs had fish-rich diets aligned with human diets, a pattern seen at other sites, suggesting deliberate human feeding practices. The study reframes the domestication timeline, proposing earlier and more complex diffusion, with multiple waves of canine ancestry shaping today's dogs. Researchers note the difficulty of distinguishing dogs from wolves in skeletal remains without advanced genetic tools, underscoring the value of ancient DNA methods. This emphasis on genetic techniques reflects the ongoing challenge of accurately identifying early dogs and understanding their relationships to humans. Experts caution that domestication likely began earlier outside Europe, and that the canine record reflects a multi-generational process rather than a single origin event, with possible interbreeding and mislabeling of fossils. A major new study in JAMA Psychiatry looks at whether giving methylphenidate to children with ADHD before age 13 affects the risk of psychotic disorders later in life. The researchers analyzed birth cohorts from 1987 to 1997, about 697,289 individuals, using instrumental variable analysis based on hospital district prescribing patterns to control for regional differences. They found that early treatment with methylphenidate did not raise the risk of adult psychotic disorders and may be linked to a reduced risk. The protective effect, however, appears to be specific to childhood-diagnosed cases and was not observed for treatment started in adolescence or adulthood. The study was led by researchers from University College Dublin and University of Edinburgh and funded by the St. John of God Research Foundation and published in JAMA Psychiatry. Experts caution that these results do not necessarily apply to other ADHD medications like amphetamines, and call for further age-specific research to understand mechanisms and potential differences across stimulant classes. They emphasize careful, evidence-based clinical assessment and ongoing monitoring, noting that while the safety signal is reassuring, more work is needed to confirm findings and to explore how results may vary by age and treatment class. GH Research reports positive Phase 2b data for GH001, an inhaled mebufotenin treatment for treatment-resistant depression, with primary results published in JAMA Psychiatry and a post-hoc analysis in Psychopharmacology Bulletin, showing efficacy largely independent of prior antidepressant failures. Correlation analyses found no meaningful link between the number of prior treatment failures and MADRS improvement at day 8 or at month 6. The JAMA Psychiatry trial showed a primary endpoint win, with a 15.5-point greater reduction in MADRS from baseline for GH001 versus placebo by day 8. Analysts remain optimistic about GH Research's potential, suggesting continued coverage and possible re-rating based on trial outcomes and upcoming Phase 3 development. Despite positive clinical data, the stock fell about 11% over the past week, signaling mixed near-term market sentiment. The press release carries forward-looking statements and notes ongoing and planned Phase 3 pivotal programs for GH001. GH Research's 2025 year-end financials show Q4 EPS of -$0.02, with progress toward Phase 3 trials after the Phase 2b success, contributing to revised analyst outlooks. Safety showed adverse events in 72.5% of GH001 treated participants, mostly mild to moderate, with no serious adverse events or discontinuations due to AEs. Limitations include the post-hoc design, small subgroup sizes, a European and white-only sample, and an open-label extension without placebo control. Authors call for larger replication. GH001 is a mebufotenin formulation delivered by inhalation, being developed to treat TRD, with a pathway toward global Phase 3 trials. Commentary from an expert clinician notes GH001's performance across patients with extensive treatment histories could benefit those non-responsive to multiple prior antidepressants. Context contrasts GH001's apparent independence from prior treatment failures with **D findings of diminishing remission with successive trials, underscoring the potential novelty of GH001. A new study uncovers a gut-brain dialogue that alters appetite during infection. Tuft cells detect parasites and release acetylcholine, which prompts enterochromaffin cells to release serotonin, activating vagal nerve fibers that signal the brain to reduce food intake. In mice lacking tuft cell acetylcholine signaling, researchers observed no infection-induced appetite suppression, establishing a causal link between tuft cell activity and changes in feeding behavior. In isolated cultures, tuft cells release ACH via TRPM5-dependent pathways, and during type 2 inflammation they also release ACH constitutively through an IL-4-induced leak-like mode that does not rely on TRPM5, indicating two distinct release phases. The findings, published in Nature on March 25, 2026, come from UCSF researchers led by co-senior authors David Julius and Richard Loxley, with collaboration from the University of Adelaide and Stuart Brierley's lab. Koki Tohara is the first author. The Nature report credits the collaboration with the Adelaide lab and situates the work within a broader context of gut-immune nervous system communication shaping behavior during infection. The study suggests that tuft cell signaling could explain symptoms of parasitic infections and points toward new treatments for gut-related conditions such as irritable bowel syndrome, food intolerances, and chronic visceral pain by targeting tuft cell pathways. By revealing a gut-brain signaling mechanism, the research implies therapeutic targets beyond infection, potentially addressing IBS, food intolerance, and visceral pain through modulation of tuft cell activity. IL-25 induced inflammation elevates FOS in vagal brainstem nuclei via enterochromaffin cell signaling, engaging a distinct NTS neuron subset from canonical nausea circuits, while acute ACH release from tuft cells activates neighboring EC cells through muscarinic receptors, HRM3 to trigger substantial serotonin release and downstream 5-HT3 receptor signaling. A new Nature study argues that extreme global climate outcomes can occur at moderate warming of 2 degrees Celsius, not just at higher levels, changing how risk is assessed. The research suggests that some extreme weather events at 2 degrees Celsius could mirror scenarios previously tied to higher warming, raising the stakes even if Paris targets are met. These worst-case results at 2 degrees Celsius arise from differences between models rather than natural variability, making extreme outcomes plausible at moderate warming. The team uses a sector-focused approach to identify where severe impacts are most likely by examining drivers like heavy rainfall, agricultural droughts, and weather conducive to wildfires. They construct spatially coherent results from single-model simulations to avoid overstating extremes by mixing incompatible scenarios, aiming for realism in projections. The study emphasizes communicating risk beyond averages and considering low probability but high impact outcomes in policy and adaptation planning. Relying solely on model averages can give a false sense of security and understate extremes, especially for staple crops and sectors vital to global food security. The findings advocate integrating extreme outcome risk assessments into climate policy and adaptation, while underscoring the urgency to limit warming well below 2 degrees Celsius. Experts urge urgent mitigation and stronger adaptation, noting that even at 2 degrees Celsius, some regions and sectors may face threats comparable to higher warming levels. The authors caution that 2 degrees Celsius does not equal the severity of higher warming, but extreme impacts can occur in vulnerable sectors, calling for risk-informed adaptation and accelerated action. Vulnerable areas such as agriculture, urban zones, and wildfire risk could face severe effects at 2 degrees Celsius, requiring proactive policy and adaptation measures. In the Iberian Peninsula, densely populated areas may experience amplified extreme rainfall and drought under 2 degrees Celsius, more than under higher warming, signaling elevated local risk. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.